Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another topic that is your introduction to the microbiology. Okay, so before starting this lecture, we have to know that what is this microbiology? So simply microbiology are defined as microbiology are defined as the study of the microorganism. Study of the microorganisms. And these microorganisms are what they are tiny organisms which cannot be seen by our naked eye. Which cannot be seen by our naked eye. That is called a microorganisms. Okay, so this microorganisms included the bacteria, fungi, protozoa, etc. Okay, so microbiology is a branch of science in which we study about the microorganisms, like which type of microorganisms are there, what is the morphology of that microorganisms, and at which temperature these microorganisms can survive, can grow. All these are come under this topic that is your microbiology. And also we are study about the different scope of that microbiology. Now let's start the today's lecture. Microbiology. Microbiology is the study of the living organisms of microscopic size. Okay, that I already told you. Microbiology are what? It is a branch of science, branch of science in which we study about the microorganisms. And these are microorganisms are what? They are microscopic in size, which means that we cannot be seen by our naked eye. For their visualization we have to use a microscope. Okay, we are surrounded by these microorganisms. Surrounded. Now, this included the bacteria, fungi, algae, protozoa, and viruses. They are what? They are the or microorganisms. Bacteria, fungi, algae, protozoa, viruses. Now, the next is your. Now, this term microbiology are introduced by a chemist, and he is what? He is a French chemist, Louis Pasteur, and he is also known as a father of microbiology. Father of microbiology. This is a French chemist, Louis Pasteur, who gave the term microbiology, and he is also known as a father of microbiology, and he demonstrated that the first time the term fermentation, and this fermentation are caused by the growth of the bacteria and yeast. Okay, now see what is written here. The term microbiology was introduced by the French chemist Louis Pasteur, who demonstrated that the fermentation was caused by the growth of the bacteria and yeast. He is known as a father of microbiology. And this microorganisms cannot be seen by the naked eye. They are only seen under the microscope, and these microorganisms are present everywhere on the earth. Okay, now if you are sitting here, now from your surrounding, there is a lots of microorganisms are present, including the human beings, Animals, plants, soil, water, food, and atmosphere. Microorganism surrounded like human beings, animals, plants, soil, water, food, and atmospheres. Microorganisms may be beneficial or harmful to a human being. If they are beneficial, then it can be used for the production of the food, and if it is harmful, then they have a capability to cause a disease into the human being. Now the next branches of the microbiology. The first branch is your pure science, and in pure science, the first one is your bacteriology. In bacteriology, we study about the bacteria. Okay. Then the next branch is your mycology, in which we study about the fungi. Then the next is your protology. In protology, we study about the protozoa. Then algology, in which we study about the algae. Then parasitology, in which we study about the parasites. Then genetics. In genetics, we will study about the hereditary and variation. And the last one is your immunology, in which we study about the different mechanism which is involved in the development of the resistance by body to infectious disease. Immunology is what? Immunology, study of the mechanism of our immune system, and the work of our immune system is what? It is a resistant by a body to fight against a infectious disease. Now the next is your applied science. Okay, in applied science, the first branch is your medical microbiology. When we study about the microorganisms which are used in a medical science, then it come under the category of medical biology, microbiology. Next, pharmaceutical microbiology, when we study about the microorganisms which are used in a pharmaceutical. Then in industrial microbiology, we study about the microbiology, we study about the microorganisms which have a application in a different industry. Then food microbiology, study about the microorganism in the production of the food, and also study about the microorganisms which spoil the food. Next category is your soil microbiology. Study about the microorganisms which are related to the soil. Agriculture microbiology, study of the microbiology which are related to the agriculture. Aquatic microbiology, study of the microorganisms which are related to the aquatic. Air microbiology, study of the microbiology which are related to the air. Epidemiology, then last is your epidemiology. This is your applied science branch. Now, first one is your medical microbiology, which deal with the study of the causative agents of the infectious disease in a human being. Okay, so in medical microbiology, what we are going to study? We are going to study about the different causative agent which have a capability to cause a infectious disease into a human being. And this medical microbiology has a clone link with other disciplines like your pathology, clinical medicines, pharmacology, and therapeutics. The next branch is your pharmaceutical microbiology. uh, it is deal with the study of the microorganisms which are responsible for the production of antibiotics, enzymes, vaccines, vitamins, and other pharmaceutical substances. So in pharmaceutical microbiology, we are study about the microorganisms which are responsible for the production of the different antibiotics, enzymes, vaccines, and vitamins, and also used in a formation of the different pharmaceutical substrate. Now the next is your, it also includes the method of the sterilization and disinfections, microbiological testing of the pharmaceutical, sterile product, preparation and diagnosis of the disease and treatment. These all are come under the category of pharmaceutical microbiology. Now the next is your industrial microbiology. In industrial microbiology, it is the we study about the microorganisms which are useful for a industry, like in the production of alcoholic beverages, vitamins, amino acids, enzymes, antibiotics, and other drugs, and also include in different fermentation technique, like the production of the different compounds. Now the next is your food microbiology, which deal with the interaction of the microorganisms and food in the relation to the food processing. We all know that these microorganisms are used in the production of in the food processing industry, and it is also responsible in the food spoilage, foodborne disease and their prevention, and include the preparation and preservation of the food product. Okay, so when we discuss about the this interaction of the microorganisms with the food related issues, like when we use a microorganism for the production of the food, and the microorganism, when we study about the microorganisms which are responsible for the food spoilage, food which are responsible for the foodborne disease, and also their prevention, and include a preparation and preservation of the food product, then this will come under the category of the food microbiology. It is the study of the soil microbes and interaction among the soil microorganisms. The next category is what? Here it is missing is that is your soil microbiology. After food microbiology, the next term is your soil microbiology. And this in soil microbiology, we study about the microorganisms which are present in a soil, and also study about their interaction with the soil. Now the next is your agriculture microbiology. In agriculture microbiology, we study about the relationship of the microorganisms and the crop, with on emphasis on the control of the plant disease and also improvement of the yield. Okay, so the microorganism which involve in a involved in a crops, what they do, they control the plant diseases and also improve the yield. Now the aquatic microbiology. In aquatic microbiology, we are study about the microorganisms which are responsible in the activities which occur in a fresh and marine water, like your lakes, rivers, bays, estuaries and seas, and also include in a water purification method, microbiological examination, and other biological degradation of the waste. Now the next category is your air microbiology, which deal with the role of the aerospora in a contamination and also in a spoilage of the food. It also deal with spreading of the plant and animal diseases throughout the air. Now the next is your epidemiology. In this category, we concern with the monitoring, control and spread of diseases in communities. So the microorganisms which responsible in the spreading of the disease among the communities, it come under that category of epidemiology, but the monitoring and control of that disease. Now, classification of the microorganisms. Microorganisms are classified on the basis of two types, that is your prokaryotes or eukaryotes. In prokaryotics and eukaryotic, the detailed videos are already uploaded in our account. You can visit and see that. Now, the next is your prokaryotic cells. Okay, here we discuss only about the basics of the prokaryotics and eukaryotics, but in detail videos you can visit our channel and see that. Prokaryotic cells are what they are their genetic material are not enclosed by the nuclear membrane. So these are the major difference between the prokaryotic and eukaryotic cell. In prokaryotic, the genetic material like your DNA, it is not enclosed with any nuclear membrane. Okay, this nuclear membrane are absent. Nuclear membrane are absent. But in eukaryotic cells, these genetic material are what they are enclosed by that nuclear membrane. Suppose that this is our genetic material and now it is enclosed with that nuclear membrane. Now the next point is your absence of the nuclear membrane. If it is not enclosed with the nuclear membrane, then obviously the nuclear membrane are absent in prokaryotic cells, but in eukaryotic cell this nuclear membrane are present. So this is about the prokaryotic and eukaryotic. Now next is your scope and importance of the microbiology. This microbiology production of antibiotics like penicillin from penicillium. Penicillin is what? Penicillin is a microorganism which are used in the production of the antibiotic that is called a penicillium, and it is also production. It is also used in the production of the enzymes, vaccines, biosurfactants, alcoholic and other pharmaceutical product, diagnosis of the disease and treatment like ELISA and vital test, treatment of the industrial waste and material, plant growth promotion, sterile product preparation, sterilization. Like sterilization means that the killing of the all type of the microorganisms. For example, moist heat sterilization, dry heat sterilization, and membrane filtration. Next is your steroid biotransformation like progesterone, testosterone. The next is your identification of the microorganisms like your morphological culture or microscopic study. And the last one is your testing of the pharmaceutical products and raw materials. So these are what? These are the scope and importance of the microbiology. Then we are going to discuss about the history of the microbiology. So we all know that Louis Pasteur are known as a father of microbiology, and he also give that coined that term microbiology. 1822 to 1895. There he was what? He was French man trained as a chemist. He is a French man and he was trained as a chemist, and he discovered the isomers of the tartaric acid and also discovered the process that is your fermentation. Okay, and also develop the another method that is your pasteurization means that heating the food product so that the pathogen microorganism will be destroyed. Okay, will be killed. And this pasteurization mainly used in the case of milk to reduce the microbial contamination of wine and beer. Okay, this pasteurization method first time are introduced in case of the wine and beer so that the microbial contamination will be reduces. And he also developed the anthrax and rabies vaccines. Okay, now the next is your germ theory of the disease, that is your Agostino Bassi silkworm disease which are caused by a fungus in 1835. Then Ignaz Semmelweis in 1841 invisible agent which cause a sepsis. Then Ricard Petri which are designed a special plate so that it hold a solid culture. This plate has great significance in a microbiology and it is referred as a Petri plate. Okay, this Ricard plate are known as what? Are known as a Petri plate which we are used in our microbiology laboratory. Then Joseph Lister, he introduced the concept of the sterile surgical field and also use of antiseptic, and also developed a limited dilution technique, and he is known as a father of the antiseptic surgery. Now the next is your Robert Koch. 1843 to 1910. He was a German physician and Pasteur rival. He studied about the disease of the anthrax and the matter so that sorry developed the matter for the identification of the etiologic agent. First, he was first man who utilized to identify the Bacillus anthracis as a etiological agent of anthrax in 1877, and also developed a staining technique and develop a set of the postulates. Now, coach postulates are what the microorganism must be present in a diseased host and absent for a healthy. Okay, microorganisms are what? These microorganisms are present in a host cell, and the host then that host cell are concluded as a diseased person and absence in a healthy person. This is a first postulate of that coach, and the next is your next postulate is microbe must be isolated and grow in a pure culture. That we are doing in our microbiological laboratory. What we do, we prepare our nutrient agar media, and in nutrient agar media we grow a pure culture. Isolate the microbes must cause disease when inoculated into a healthy laboratory host. Must re-isolate the microbe from the diseased laboratory host. They are what? These are the coach postulates. Discovery of the chemotherapeutic agents. Paul Ehrlich, he is known as a father of the chemotherapy. In 1930, he introduced a drug, salvarsan, for the treatment of the syphilis. And in 1929, Sir Alexander Fleming accidentally discovered a substance which is known as the Penicillium notatum. He extracted from the fungus a compound which is called a penicillin. This penicillium are extracted by this penicillium, which is a fungus that that could destroy several pathogenic bacteria. And in 1944, S.A. Waxman discovered another antibiotic that is known as a streptomycin. So this is all about the introduction of the microbiology or basic of the microbiology. I hope you will understand, but if you have any query related to this topic, you can freely to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and um, subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also visit our website, that is www.itlsacademy.com, and ask your query to the helpline number that are already mentioned in your slide, that is 7080833450. Thank you for watching. Please like, share and subscribe our channel.